but there are over 40 studies that demonstrate that dietary iron deficiency in toddlers between 6 and 24 months, so this is usually dietary acquired, that they have behavioral abnormalities while they're iron deficient, including motor and cognitive problems, and that the cognitive delays last for 19 to 23 years afterwards. So these babies are these children are treated, and Betsy has been able to follow up these cohorts, and the types of problems that they seem to have are characteristics of striatal problems, dopaminergic things, frontal problems, and some hippocampal dysfunction. The electrophysiologic abnormalities include delayed myelination, things that we think are associated with delayed myelination. That is, longer latencies on auditory brainstem responses, both at the time of iron deficiency and 2 to 4 years after treatment. And this would be characteristic of the myelin problems. So I talked to you about energy problems, myelin problems, and monoamine problems, and I think we see those in the postnatal dietary iron deficiency. The neurobehavioral follow-ups of perinatal iron deficiency is much less. There are many fewer studies, but the behavioral abnormalities include that in preterm infants who are in the lowest quartile of ferritin concentrations have far more abnormal neurologic reflexes than those with more adequate iron status. Term infants who are in the lowest quartile for iron stores have poorer school-age neurodevelopment. And iron-deficient infants born to iron-deficient mothers have altered temperament right from the beginning. You can see this has been quantified by psychologists in the immediate newborn period. From our laboratory, and I'll show you some of the data from this, we know that infants of diabetic mothers who are iron deficient have impaired recognition memory processing. And of course one can ask the question, is it the diabetes or is it the iron? And I'm hoping to convince you that in part it's about the iron. So how do you assess memory function in a newborn infant? And why would you want to do this? One of the reasons you want to do this is that when you are talking about a nutritional deficit that is affecting brain function, you don't want to wait 7 years to do your assessment because many things can happen in those 7 years. So the better your brain assessments can be and the more precise it can be, and we'll talk about this more in the second lecture, the more you can say, yes, that was due to the nutrient deficit. And we use something called event-related potentials, which are predictable components in the EEG. These are microvolt changes, not millivolt changes like you see in a regular EEG. And we look at it over time and index it to memory events. So we have a baby do a memory event, and then we record, and we look at the pattern of EEG processing. And this is how it looks. So this would be one of our newborns. And what I've done here is I've put the mother saying, "Hi, baby," because that's what she says to the baby, in red, and that's the color it'll be on the graphs. And we compare that to the baby's response to a stranger saying, "Hi, baby." Okay, and that will be in blue. The stranger, by the way, is just the mother in the next room. But the assumption here, and actually it's been shown behaviorally, is that babies recognize their mother's voice on the first day of life. Okay? We now have electrophysiologic evidence that they also recognize dad's voice, for those of you who are worried about this. But they certainly recognize mother's voice, and there is this very characteristic footprint or electrophysiologic response. Now when the children get older, you can put on as many as 128 leads, and you can even tell from the analysis of those leads, the vector analysis of those leads, where the signal is coming from. So we can actually identify that it's coming from hippocampus. But in the young baby, we just use these leads to look at patterns. Okay? And this is what we see. And here is the control baby. And these are different. This is from the posterior, this is central, this is frontal. These are all from the midline leads. And you can see that there's this deflection upwards for each of the two stimuli, whether it's the mom's voice or the stranger's voice. And that's just attention. That means you've got the baby's attention. The baby's actually hearing and starting to process. But you notice that with the mother's voice, the red line, that the line comes back down to baseline. That means that no further processing is needed. The baby processes the mother's voice very quickly, presumably showing that it recognizes it. It no longer processes it over time. But with the stranger's voice, the baby actually continues to process with this negative slow wave for up to 2 seconds afterwards. Okay? And that's the baby updating or encoding a new memory. Babies have a natural desire to look at or listen to novel stimuli. And that's what we see electrophysiologically. When we look at a wide group of infants of diabetic mothers who are both iron deficient and iron sufficient, so this is a mixed sample, we see much less discrimination than we see in the control baby. This is not significant here. And when we differentiate the brain iron deficient infants of diabetic mothers, so these are the ones with the ferritin less than 40, these are the ones with the ferritins more than 40, you see nice differentiation between mother and stranger, difference between the lines, and very little in the brain iron deficient ones. Now, short of going in there and asking the mother, may I now biopsy your baby's brain to prove to you that your baby is iron deficient, and our institution won't let us do that, we now need to work in models to try and figure out what is going on. But what's interesting is that when you bring these babies back when they're 3 years old and do the high density array, we see that their brains are really organized very differently. That is, these are hot and cold maps, that is whether electricity is running toward the electrodes or away from the electrodes. The control baby has a different map for familiar versus novel. We expect that. So does the infant of the diabetic mother who was iron deficient. But notice how differently wired their brains are. The patterns don't look at all the same. So what that tells me is that during that critical period of late gestation when these infants of diabetic mothers became iron deficient, because all that iron was going into the red blood cells, that it caused the brain to be wired differently. Oops. And how might that be different? Well, we're going to go into the animal models now, just very briefly. This is the hippocampus in a rat. And this is the area of the hippocampus called CA1, where memory formation occurs. And there's a complex glutamatergically based set of events with signaling molecules that change the structure of the dendrites and their electrophysiology when there is a memory event occurring. The rat, by the way, has a much bigger hippocampus for their brain than humans do, which is why they can spatially navigate so well. The arborization of the neurons in this area called CA1 is highly dependent on adequate energy status, which we think is compromised in iron deficiency, and adequate amounts of glutamate, which is the neurotransmitter that stimulates there. And both of these we can show you are compromised by iron deficiency. The result of which is, so now this is the area CA1, and these are the cells that are there, and these are what dendrites should look like. We've put a marker on here looking at microtubule-associated protein. This is a protein that provides the scaffold for these neurons as they are branching and differentiating. You can see that in the iron sufficient, these are beautiful. It looks like red rain, just nice long dendrites ready to accept axons and make synapses and have activation with this compound called alpha-cam kinase 2, and you see a lot of activity in CA1. In iron deficiency, and this is by the model induces a 40% decrease in brain iron, the same as the babies. That's why it was important to know those autopsy studies. We see a tangled mess in here. We see very altered dendritic structure. We see very little activation of the signaling compounds, cam kinase alpha. And so we think that structurally these brains are really quite different. When we look genomically, it turns out that iron deficiency also has effect on the hippocampal genome. affecting cell adhesion, localization, signal transduction, which is important for the efficiency of the hippocampus, and just primary metabolism, so there's the energy effect. When we look long-term, these animals are now adult. It takes them longer to learn, that is, it takes more days to reach criteria. When you have them learn a task either with a 5-minute delay or a 15-minute delay, iron-sufficient animals take fewer days to learn the task than iron-deficient animals, and the animals continue to have changes in their dendritic structure, changes in the electrophysiology, lots of scar formation with astrocytes in the hippocampus, and still changes in the metabolism. So long-term changes, even though these hippocampi now are completely iron sufficient. Okay, I'm a little bit over, but I want to take a little time to go through dosing. The current AAP recommendation, American Academy of Pediatrics recommendation, is that for preterm infants, we should be giving them 2 to 4 milligrams per kilogram per day of enteral iron. The general rule of thumb has been if the babies are less than 30 weeks, and the more premature the baby is, the higher the requirements would be. Again, because they did not accrete all of the iron that they would have prenatally. So we use 4 milligrams per kilo per day if the babies are less than 30 weeks, and we used more like 2 to 3 milligrams per kilo per day if the baby is premature but greater than 30 weeks. Now remember, there is the risk that these babies, the less than 30 weekers, are also antioxidant compromised. So you're giving a higher dose of iron because the requirements to a baby who may not be able to handle it quite as well. If babies are on EPO, and our unit does not use EPO, then 6 milligrams per kilo per day. Post-discharge, the recommendation, or what is actually in the post-discharge formulas, provides 2.25 milligrams per kilogram per day when the baby is feeding 150 ml per kilo per day. And it looks from our data that I showed you that that may need to be increased to as much as 3.5 milligrams per kilo per day. We have started, and this is not national by any stretch of the imagination, but I think one should consider monitoring the ferritin level at birth, at the time of discharge, because there's a wide range of iron status at the time of discharge. If you have a baby who's been multiply transfused, they may have ferritins of 500. Or if the baby has been a pretty benign baby who grew very rapidly, that baby may have a ferritin of 20. And it has implications. So we think that we should be monitoring the iron status a little bit better, more completely, at the time of birth, at the time, throughout the hospitalization, or at least at the time of discharge, and then after discharge in the follow-up clinic. So in summary, I hope I've convinced you that iron has an important role in the late gestation and the early postnatal brain development, that different structures at that time might be vulnerable compared to later iron deficiency, and that iron deficiency presents a risk to the developing brain that seems to be, at least in our models, long-lasting, even after you've treated, that there are critical periods that are dependent on iron sufficiency. The abnormal behaviors that we see with iron deficiency do indeed map onto or are emanating from those structures that we see affected in the models, so the brain regions that are at risk, and that our current iron dosage recommendations in the NICU are probably pretty adequate. I don't think that would change what I do, but I think they are probably too low at follow-up, and that we should be concentrating a little bit more on assessing the iron status of rapidly growing or babies who are doing catch-up growth in the post-discharge period. I don't actually do the work. I just talk about it. These are my many friends and collaborators throughout the country and the people in my laboratory that help me with this. Thank you so much for listening.